So it's my pleasure to introduce the next speaker, who is Rieko Ishima. She's at the University of Pittsburgh Medical School, and her pedigree traces back to Dennis Torsha at the now very famous NIDDK building, where there's all these folks there. Anyways, Rieko.
Hello, my name is Rieko Shima. Thank you very much for the introduction. And I received a PhD in Japan, Akasaka. Yes. And today, initially, I was planning to talk about this conformational change in HIV reverse transcriptase maturation. However, last night, when I listened the roundtable discussion, there were two important things. One is such an integrated structure biology and NMR role. Definitely, this may fit to that aspect. But also another one was high field NMR, super high field NMR. And then the other general interest, today I want to spend initial half of my time to discuss our previous work for radiation damping for large molecules. And then next, I want to talk about this RT maturation study. And the first one, this one, story is the following. I moved to University of Pittsburgh, 2006. At that time, for me, it was the first time to use 900 megahertz NMR. I mostly used 600, 500 megahertz NMR, but not such a super high field. And then I start to look at the spectrum. And initially, we want to check what's going on, how much sensitivity is. And together with that, I start to be interested in the radiation damping effect caused by the abundant hydrogens, abundant spins. And of course, mechanism is known, theory is known, experiment is known, but this is more like to know what's going on in our NMR instrument. And the first experiment I show here is just apply pulse and then detect signals. Here, a little bit bump is the that radiation damping effect, that inverted magnetization, going back to the Z by that radiation damping. It was very fast, within 5 milliseconds. And then I had a very stupid idea. How about just add field, pulse field gradient? Now, this is a simple mind, right? Once we apply field gradient and residual magnetization gone, we may not see anything. But in fact, what I saw was this, real and imaginary, boom. Basically, although field gradient dephase residual transverse magnetization, after that, small amount of XY component generate still enough to generate radiation damping and bring back magnetization. That's why in transverse magnetization, such as you can see huge intensity. By doing this experiment, I decided to study more about this radiation damping. And as I mentioned, all theories are quite well known. And also there are also papers describe such a radiation damping for cryoprobe too. And initially I did the simulation to understand what kind of things happen based on this Warren-Warren's paper. This is a simplified equation, doesn't contain phase effect, which is included in the later paper by other group. And I will explain these two simulation results using the next slide. Firstly, when we flip little bit magnetization, and then magnetization go back to Z by degenerated, because there's transverse magnetization, which generates radiation damping. Because bulk magnetization is enough to generate the electric current to bring back magnetization to Z. And then the XY, the small magnetization, the sign of the radiation damping is going down, decrease transverse magnetization. It gets to 0. That's nice. However, it is well known that once you flip, like close to 170 degree, and then Z magnetization going back to plus Z, and X magnetization, initially the sign of this magnetization recovery is going increase the transverse magnetization, and then upper hemisphere decrease the magnetization. Therefore, you will see this increase the magnetization and decrease, and because of that, your signal overflow by the water molecule, water signals. Your FID is overflow by the water signals. And just to understand that, experimentally, this phenomena, I did a nutation experiment. Means that as written in this bottom figure, apply B1 and detect, and increment B1 little by little. And here the graph, final data I received is I could receive was a kind of FID-RS, similar to the 2D experiment. Increment, duration of nutation, and then recorded data. For each, something like spike indicate very, very short FID. And we don't, and this presentation doesn't tell at which timing radiation damping happens or which timing intensity is highest. But anyway, by doing this experiment, initially transverse component increase, that is this one, and then the sign changes after 180 degree, and that's quite expected. Then I did similar experiment, but try to detect residual Z component. Means after nutation experiment, apply field gradient, dephase entirely transverse magnetization, and then little bit flip angle will give the projection to the XY that we detected. That's also sigmoidal, that's nice. And of course, once the, after 90, there's a kind of magnetization going back to Z, therefore you see the radiation damping effect. What I wanted to show here today is the next experiment. Up to here, I pretty much understood. And this I saw funny. And initially I didn't understand why I saw this one. Here the idea was, again, nutation experiment, apply field gradient, and the magnetization is supposed to be all dephased, but we don't see any signal. And however, that was true in the initial 90 degree, but wasn't true when nutation goes. What is nice is I can rotate this one. Once it gets to this lower hemisphere, basically we saw this radiation damping effect because the sign of transverse magnetization increased in this lower hemisphere and then decreased in the upper hemisphere. And then in addition to that, there was some phase effect. This is what I try to mention here, this one. Because of this, when the water magnetization gets to lower hemisphere, we see increase of the transverse magnetization. That is a bad thing. And that is this part. And based on this experiment, we modified, at that time we modified simple HSQC experiment. For example, here there's a proton 180-degree pulse, and not to flip water magnetization. At that time we used reverb pulse. But if we use reverb pulse, which only invert NH region, but not water region, typically we needed a very long reverb pulse. But the idea which I got from the previous experiment is, in fact, we can hit to the water signal. We can perturb water signal as long as water does not get to lower hemisphere. And because that such an upper hemisphere, water magnetization just decay to zero anyway. And I call that this is a hemisphere concept. Anyway, it's similar to water flipback, but what we thought is we don't need complete water flipback. To some extent is enough. Then later on, radiation damping bring back to water magnetization plus G. And when I had started to use 900 megahertz NMR, I was playing with those things, try to understand what's going on. And then later on, I start to use those magnets for our biomolecular NMR application studies. And here now, I will switch the gear to this HIV1 reverse transcriptase. And this is an enzyme in human immunodeficiency virus 1, human immunodeficiency virus 1. And typically, it is known its function relatively soon after this virus entry to the cell, where viral RNA is reverse transcribed by this enzyme, RT. And this is enzyme is a matured enzyme. And for here in this step, the structure function inhibition of this enzyme has been well studied last 30 years. However, this enzyme is initially expressed as a polyprotein in this late stage of this virus replication. And structure studies of this enzyme in the late stage is getting to be important by two reasons. One, we don't know the structure, or biophysically we don't know. And second, as a reality, since there are many good drugs, HIV drugs exist, patient can suppress virus production. But at certain point, that DNA, virus DNA is waked up. Therefore, it is important to stop the replication in this step too. That is the reason why we are interested in this later stage of RT. And about that, I will tell you a little bit this, what was a mature RT, what's the problem there, what is a precursor, our NMR data. Because of the time, I don't include but such a consistency with other structural biology data. That is important part. And here, matured RT. This is an enzyme initially expressed as a part of polyprotein, GAG polyprotein, encoded as a 66 kilodalton protein. Its matured form is known that the heterodimer with this P51 subunit, that is 51 kilodalton protein. And this P51 kilodalton protein is generated by the cleavage at this site, and the processing is done by HIV protease encoded here. And we often call this cleavage site as a P51 RNSH cleavage site. and one RNA domain is cleaved out. But once we see the structure of the heterodimer or isolated RNA, whichever the case, this processing site is, in fact, little bit inside of RNA domain. It's a beta sheet site. Therefore, we know that we can, in the biology, we can, or structural biology, we can speculate that this site is not is buried, therefore protease may not be able to access the site easily. And of course, if the site is exposed to solution, protease will easily cut that site. That is why, and however, this is end product structure and not the precursor structure. That is why we are interested in this precursor structure. And based on biochemical virological studies, it is known that dimerization of this P66 region is essential for this RT maturation. Therefore, our immediate interest is this P66 homodimer. Total size is 130 kilodalton. And of course, it's not very huge in general structure biology, but very large for NMR, solution NMR experiment. And not just size, the field is a little bit complex because currently there are 3 models for P66 homodimer has been proposed. Right-hand side, our model, and here we proposed P66 forms symmetric homodimer with folded RNA domains. Of course, symmetry we mentioned is a symmetry within this NMR time scale. Of course, we know there's a dynamics in solution. Second model proposed was proposed by Bob London's group, and they proposed asymmetric dimer. One of the 2 RNA domains is unfolded. They didn't do experiment of actual processing, but they proposed that this unfolded domain may be cleaved by HIV protease. The third one more recently proposed based on EPR by Maria Skrua's group. Asymmetric dimer, but both RNA domains are folded. Since EPR experiment is done in the flash freeze condition, if there's motion, it may end up to the asymmetry. And about that, it may not much difference from our model, but the unfolded model may be different from ours. But what I want to discuss today is not which model is correct or not, but not just one point structure, but try to understand the structure, whether those explain functions and or mechanism of actions. For example, once ligand binds to the protein and activity changes, we compare structure with and without ligand bound form, and then we can assess the structure whether that explains the function or not, something like that. And for that, we used this maturation experiment of RT, because we know that this homodimer is cleaved by protease and gets to heterodimer. However, we found that this processing rate here against the time is enhanced when tRNA is located here, add to the solution. And here the P51 production is more significant compared to in the absence of tRNA. And here I said tRNA, and we use lysine 3 tRNA because it is known that this tRNA together with viral RNA interact with RT to form reverse transcription initiation complex. And this tRNA comes from multi-amino acid tRNA synthetase. A professor in Ohio State University studied this very well. And when this tRNA is released, it is released in the form of this lysine tRNA synthetase. And this lysine tRNA synthetase is also recruited to the virus together with tRNA. Well, we did knock down, our collaborator did knock down experiment. And as I shown in this top gel, P66 was accumulated. Although in cellular environment, there are various kinds of DNA, RNA exist. This may suggest that the tRNA may help this RT maturation for the virus replication. Unfortunately, we couldn't get difference in the RT, but we didn't see any abnormality of RT maturation in the virus in the knockdown experiment, because knockdown basically killed the virus, and this is only those escaped from the knockdown. Anyway, we had such a reasoning to use this assay, and then initially we compare NMR spectrum with and without tRNA. Here is the isoleucine methyl NMR, and here is HSQC with and without tRNA. And we saw some signal intensity changes, particularly in this random coil region in the methyl data. And in the HSQC data, orange marked ones are residues from RNSH domains, and the green marked ones are from the thumb domain in P66. And upon tRNA binding, these thumb domain signals decreased, and some of RNSH domains still observed. However, in this experiment, we don't know which subunit signals are changed in the dimer. And because of that, we decided to distinguish two RNS domain signals by introducing inhibitor to P66, NNRTI. And because in previous experiment, right-hand side, this square rectangular box, previous experiment with Angela Gronenborn, where we labeled this P66 with fluorine at residue 181. When inhibitor was saturated to the protein sample P66, we saw only two sets of resonances, right-hand side, and this peak pattern was explained by the overlay of the heterodimers. Labeling was in the 66 subunit, labeling was in the P51 subunit. That means this site, we know the stoichiometry, one inhibitor binds to homodimer, and environment changed here, at least for residue 181. And we used this node for inhibit, and then inhibitor to split RNSH signal. We could do that because this tRNA effect for maturation was not affected by inhibitor so much. Little bit helped, but still tRNA effect was much more significant. And next is a data for this right-hand side, P66 plus minus inhibitor. Now the signal split to subunit for two subunit, and then at the tRNA, there were additional signal changes. And all of these I summarized in this for the residue 434, where that is located close to this processing site. And like this, yes. And here, this site residue P66 in solution, we saw only one set of signals, and because of the molecular symmetry in solution, and upon inhibitor binding, now we see two signals. But this is not the form that, even if we add protease, this is my timer, protease, maturation is not enhanced. Maturation is enhanced quite significantly when there's a tRNA. At that time, in fact, one signal positions were gone, and the other still at a similar position. Over here, B stays. And in fact, this is a position relatively close to that in the heterodimer. Or maybe this is RNase domain protected upon this maturation step. Well, here I mentioned how that the conformational changes happens based on our data and in vitro maturation assay. What we observed sounds reasonable, but then this part is important. This is still within the NMR data, what happens if we see overview of the protein. And for that, we started SAX experiment. Last night, I decided not to show the data because our data is not nice compared to Walter Chagin's data. And anyway, those things are important. And I thank to the people in my group and the collaboration with Angela. Particularly, most of the part was done by a graduate student who received a PhD and now in Emory, and he was in this MBSB graduate program. Please tell about this program to your students and colleagues. Thank you very much for your attention.
Okay. Again, we'll give some time for the questions to build up, and I'll start. So my first question is a very simple one. When you bind this drug, I guess, why do you only get one binding to the dimer? Is that lucky or?
That's a good question. And we tested several similar series of drugs. In fact, that part was done by Angela's student. Oops. And it's likely that once the inhibitor binds to one site, structure changes happens, and that triggers structure changes of next subunit, and final conformation likely similar to the heterodimer. But when the inhibitor is smaller one, in fact, there are inhibitors which binds to monomer to both. But this particular inhibitor binds one subunit that triggers structural changes, second one gets to another conformation.
Okay, so it's anti-cooperative. Yeah.
Very interesting, yes.
Yeah. That is interesting.
Mm.
My other question works backwards to the first half of your talk, and that was, so you showed that you could use the effect of radiation damping in the HSQC experiment instead of giving the pulse. So have you now, is this sequence now what you always use for your HSQC?
Not always, only when we want to excite like NH part. Okay, I will say that I used T2 dispersion experiments that I was doing before very frequently because I worry about that amide water exchange. For regular HSQC, we don't necessarily use, but probably when, like for example, T1 period becomes very, very, very long, and when we don't want to apply very, very long gradient, this may be still useful.
Yeah, so for like disordered regions of proteins or, you know, those unfolded proteins that will exchange.
I see, that may be interesting point. Yeah. That may be interesting point. Yeah. Since we don't study unfolded protein much, yeah, we don't know.
Okay, I guess you blew people away as far as I can tell. Those were my two questions.
May I ask a quick one?
Please.
Since you've been there at the mutation or the radiation damping section, when you showed the slide of the mutation experiment, the imaginary part at the null transition had some pretty substantial spikes. Are these artifacts or is that something else?
Which one?
The mutation experiment.
I had the 3 mutation experiment.
This one here, yeah.
This one, okay. This one is...
in the imaginary part.
Yes, yes. Basically, when I mentioned here, we did the simulation. This one is using this paper, Warren Warren's paper in 1989, where phase is simplified. Phase component is included. And in fact, the phase modulation happens. Depends on what magnetization remain in transverse, and also that generate radiation damping. But it's not only one direction and the axis, it goes to a perpendicular component too.
Right, but the peaks or the measurements right at the null transitions for almost every other period seems to have these huge signals.
Yeah, when this is based, okay, or how about, for example, in this case, like, it is true that the one half is very small and the half hemisphere is large. Maybe this is also maybe difficult to see this one. Yeah, half hemisphere, this gradient effect is large because even it says 90 degree or 180 degree, there's some residual magnetization exists, not only certain X component, but the Y component too. Mm-hmm. In fact, this intensity also depends on shimming too. As you can imagine that shimming make residual water magnetization that affected to this experiment too. Mm-hmm. Yeah. That's a good point.
Yeah. Okay. Thank you.
Thank you very much. Our next speaker, that's Steve Van Doren from the University of Michigan. His lineage traces actually through Eric Zoidevec at Michigan back to Kurt Wuthrich. Steve. Oh, we see your slides, but we don't hear you.
I got unmuted.
Okay.
Greetings from the other side of the Gateway Arch, actually in Missouri. I would like to thank the organizers and our hosts and the presenters for a fascinating program. I've really enjoyed it. I appreciate the invitation to join you to tell you about our NMR journey from structures to inhibitors of transient interactions of the proteolytic enzyme called MMP7 with fluid partners from cell surfaces. Now, this is an enzyme put on the map at Vanderbilt by 2 decades of work of Lynn Maturation, in the context of cancer especially. So here, this very soluble enzyme is shown bound to the endometrial lining of the uterus. And so I was really intrigued by the question of how this soluble enzyme sticks to cell surfaces. And MMP is referring to the metylated proteinases out in the extracellular matrix. Now, it has healthy roles in the lungs and governing wound healing, and also in the health of other mucosa, including in the gut, where it activates the antimicrobial defenses. But it's, like I said, it has unhealthy roles in promoting invasion and aggressiveness of tumor cells in epithelial cancers of breast, uterus, pancreas, colon, and melanoma, and others. And so these are the former Vanderbilt crew that did a lot of this work. Now, I'd like first to consider the membrane proximal activity of MMP7. There are 2 mechanisms of cell binding, and that was known for a long time now, to lipids and to glycosaminoglycans, or GAGs for short. Now, these are and I should mention that there are sulfated lipids found in lipid rafts, such as cholesterol sulfate and a sulfoglycolipid that recruit MMP7, which makes tumor cells aggressive, as pointed out by a group in Japan, Higashi and Yamamoto. And this promotes cell adhesion, and it promotes MMP shedding of proteins from epithelial cell surfaces. Now, we've already had good introduction to bilayered micelles, bicelles. So we've made them simply by taking DMPC liposomes and dissolving them with micelles. And Chuck Sanders, I appreciate Chuck Sanders' lab for advising the world to use short chain lipids that have a lower critical micelle concentration to have fewer free monomers. And I have to acknowledge the caveat about these low Q bicelles, referring to Q equals 0.5, that is 1 DMPC to 2 short chain lipids. Linda Columbus, Peter Thielemann, and Glover, they all warned us that these may actually be forming mixed micelles, and that is that the short chain lipids that we thought were mainly on the rims may actually mix into what we thought was mainly a bilayer. So they're not so discoidal as we would like to have thought. Now, our strategy is to exploit the NMR sensitivity to transient proximity by spin labeling the lipids or carbohydrates in order to measure PREs. Because, of course, these are a dipole-dipole interaction that are highly sensitive to proximity. And we've been using this type of information to strain molecular dynamics simulations to fluid models of the bilayer or with a carbohydrate chain. And so, as you know, the PREs depend strongly on the proximity. So if our partners are far apart, then we expect to see full peak heights. But as our partners, you know, the site shown in blue, it approaches the spin label, which is in the membrane. In this case, we see our NMR line, our proton lines, that is shrinking away with that proximity. Now, we like to measure that as a exponential decay for accuracy, and in order to get that into gamma-2 relaxation rate constant, which we can relate together with the rotational correlation time to the distance r between these sites, or, you know, our approximation of r when it's actually complicated. Now, Steve Pryor did this with the MMP, proform of the MMP7 with bound to zwitterionic bicelles or to bicelles made anionic by cholesterol sulfate being dissolved in them. And so the catalytic domain is the bigger sphere and the pro-domain is the smaller sphere, and the grid represents the average plane through the phosphoryl groups of the head groups. And you can see that there's a transient binding to the zwitterionic bicelles, or sorry, superficial binding, and a deeper and reoriented binding when cholesterol sulfate is present. And so we took our NMR structures that he had done with 3,000 NOEs and these revisions of those NMR structures with the 70 distance restraints across the interface, or 120 on the right. And he created ensembles out of these. And in the MD simulations, you could see that the lipid head groups were organized in the interface in the zwitterionic complex, and more so in the anionic bicelle-bound complex, where there was partial insertion. And in fact, these lipid head groups are pushed out away from the protein, creating a couple of little bowls, and they're really quite misshapen lipids as it inserts. Sorry, that's on the right. We have cholesterol sulfate present. And this is another portrayal of the same thing, where you see this reorientation from the zwitterionic state on the left to the anionic state on the right. And there's an 80-degree rotation or so and roughly an 11 angstrom insertion. So we see 10 more NOEs and I guess 50 more PREs in the case on the right. And also at the bottom, for the peripheral association with the zwitterionic bilayers, we have tryptophan 129, which is this one here within the box. It's sitting superficially on the head groups of the choline head groups, whereas it's inserted into the bilayer down here and having lots of NOEs to the acyl chains in this inserted state. We also have cholesterol sulfate present in this bilayer. Now, that was all done with an inactivated proenzyme or zymogen, whereas this experiment on allostery was done with an active zymogen. And Steve and another postdoc in the lab found that when they incubated this active zymogen with bicelles, it actually activated from the full-length proform to the active catalytic form here at the higher lipid concentrations. And so whereas in the free state of the zymogen, we have cysteine coordinating the zinc to keep the enzyme inactive. We have a salt bridge here. The NOEs indicated a reorganization, indicating that the cysteine is moving away and the aspartate is moving here. And so this was a real surprise for us to see that there was allostery triggered by the bicell association. And so a displacement in the coordination environment. Steve also found that there is a network of, when you plot the location of the chemical shift perturbations from the addition of bicelles and bicelles with cholesterol sulfate, propagating from that key tryptophan that inserts into the bilayer around the periphery of the enzyme toward the active site. So he could partly account for the allosteric effect that I just mentioned. Now, the MMP7 binding to glycosaminoglycans at cell surfaces is important for the killing of bacteria in the gut by activating and shedding of pro-alpha defensins from the crypts in the intestine to kill bacteria. MMP7 also activates neighboring pro-MMP7 molecules. MMP7 is also, its glycosaminoglycan binding is important in its healing of lung wounds and in the remodeling of, in postpartum mothers of the uterus and mammary ducts. And this occurs by the activation and shedding of pro-HB-EGF, that is the maturation so they can bind the EGF receptor, which is portrayed here by Stamenkovic's group 18 years ago. This is a heparin sulfate proteoglycan called CD44 with GAG chains emanating from it, which bind to the pro-HB-EGF and to the MMP7, which is here symbolized as this pennant, which can draws the protease and the pro-HB-EGF together so that proteolysis can occur to mature it to HB-EGF, which can then bind to the ERB4 receptor, which is so important in female biology and in cancer progression. Now, what sort of carbohydrates did we use for our studies? We used heparinoid chains of the length of 8 residues derived from heparin as a model of the physiological chains of heparin sulfate. These, especially the heparin, are heavily sulfated, but also containing carboxyl groups. So they're very anionic. And because they're so anionic, they tend to be very extended, but they also have this undulating structure that you can see here in these models. Now, there is, well, let me back up and say that there are chemical shift perturbations that occur in the pro domain here, which are expanded up here in the inset, and also in the catalytic domain. The pro domain CSPs map to these yellow colored residues, and those in the catalytic domain to these blue colored residues. And this was in physiological saline where we see a 70 micromolar KD. Now, given that we have so many basic amino acids that actually circumscribe nearly the entire zymogen, and we have lots of small CSPs and few NOEs to the glycosaminoglycans, how do we go about discriminating among the potential contacts with these many, many basic amino acids? So we replaced by site-directed mutagenesis the basic side chains that we found to be near backbone CSPs. And we found 3 patches to be important in heparin-triggered maturation of the enzyme. Firstly, this carboxy-terminus region, which is a polybasic region. Secondly, the back of the catalytic domain here. And thirdly, this residue in the pro-domain. And we figured this out, that is Jan Fulcher figured this out through these maturation assays that are triggered by the presence of low molecular weight heparin. And so you can see there's a very easy and ready activation of the wild type zymogen with just a little bit of heparin. But that is really diminished when we remove the C-terminal 6 or 8 residues. If we only make a single point mutation, we get a small diminution of the activation. But if we do both a point mutation and the C-terminal deletion, we really impair the activation tremendously. Okay, so these the time courses of these assays change from the wild type situation here to situations up here as we mutate important sites. Okay, so we're getting a functional ranking of our NMR results that way. Now, we also used two kinds of paramagnetic NMR mapping of the weak and transient contacts, both of which are generalizable. One is to spin label one partner, in this case, the glycosaminoglycan, and the other is to spin label the solution, which, as you know, is called solvent, leads to solvent PREs with which we can make footprints of the binding sites. Now, the spin labeling of the 8-mer chain, which we call DP8 for degree of polymerization of the heparin chain, this spin labeling was done at the reducing end by Bob Linhart's group at Rensselaer. And this, when we mixed it with the Zymogen, we got PREs all over the place throughout the chain. Now the prodomain is looking much more prominent than it did from using the CSPs. These PREs emanating from the reducing end strongly suggested to us that the reducing end is inserting into a cationic pocket in the prodomain here on the left and into a cationic pocket along a cationic cleft on the back of the catalytic domain on the right. Now, we also did this paramagnetic footprinting using differences in solvent PREs. Here our snowman is representing the Zymogen, and we have a basic site, and we incubate, we collect our TROSY, and then we incubate with a gadolinium chelate, and we see the surface amides being broadened away. Then we introduce our heparin chain, which in this case was only 4 residues long. And then we do it again, we see, well, it may or may not have changed depending on whether that heparin chain was binding. We saw large protections in the prodomain, again, implying that, well, okay, so we saw a big diminution of the PREs in the prodomain. And so this is the free state, these two peaks. This one was very sensitive, this one to the gadolinium, but once we added the DP4, it was less sensitive to the broadening by the gadolinium. So we saw big protections here in the prodomain, but also in the catalytic domain, again, sort of giving us a different picture than the CSPs did. Now, and in general, I would say over a 25-year period, I found that paramagnetic methods have seemed to be more accurate than CSPs have. I would make that a pretty strong claim. So we see the paramagnetism highlights the binding in the prodomain, as I mentioned. And so we have both footprinting results of the yellow sites, and we have the PREs from the tethered sites in the heparin chains in the blue, and we have both at the green sites, both in the prodomain at the top and at the back of the catalytic domain here, which is in the foreground at the right. Now, how do we deal with this in structural modeling? Well, we divided and conquered, and we simplified it into 2 modes of binding. One which is bridging between the pro and the catalytic domains on the left, and the other which is binding to the back of the catalytic domain, and which is better defined by the PREs and also by the site-directed mutagenesis. Okay, and we noticed that this basic site on the back of the catalytic domain coincides with the same basic site that associates and inserts in anionic bilayers. We decided to target part of this GAG and membrane binding site for drug discovery. Atom-wise screened a virtual library of 6 to 7 million compounds using their proprietary AI model. And they awarded us 72 compounds which have diverse scaffolds, and we then screened them by ligand-detected NMR, and we found that 22 of them bind using this T2 relaxation-based assay, in which the very slow decay of the compounds was accelerated in some of the cases, such as this example, by the presence of like a 10% presence of the enzyme, so like 100 micromolar ligand and 10 micromolar enzyme or so. So you see a more rapid decay in the purple curve at the bottom. We confirmed this using stacks of TROSY spectra, that is one liganded spectrum for each of the 22 compounds, confirmed that there are CSPs consistent with all 22 of these binding, and we could rank them relative to the free unliganded state over here using our Trend NMR Pro software. And we could also measure the affinity of selected titrations, that is, titrations with selected compounds. We had this is an example of an inactive binder, an allosteric inhibitor, and an allosteric activator. And this is using our Trend NMR Pro software, in which we take the stack of different liganded TROSY spectra, and we do PCA, and we can rank them, or we take a titration and we could fit it as shown here based on the PCA. Okay, so now, yeah, admittedly, these compounds are pretty modest in their affinity. Okay, I acknowledge that. That's an important point. And we saw, in fact, a range of allosteric effects on the heparin activation of the pro-MP7. So in this gel, we see only that compound 9 is inhibiting the activation by low molecular weight heparin, and that's at a very high concentration of about 1 millimolar. Whereas compound 11 is actually promoting the activation, and compound 8 also, but to a lesser degree. And actually the majority of the compounds promoted the activation, okay, which was interesting. I'm trying to advance, there we go. So, and surprisingly, the biggest of the CSPs did not bind our targeted pocket, or we're not mapping there, but rather over on this side of the catalytic domain. And so there's, you know, it looks as if this could be a novel allosteric site that all 22 of the compounds are binding to this arginine and to a tryptophan next to it. You know, we have to say, oops, the virtual screening didn't work like we expected because we're getting binding at a different pocket. But on the other hand, 10% of these compounds are inhibitory. And the others are quite interesting themselves in activating the enzyme. So I've shown you that both anionic GAGs and bilayers can recruit and activate MMP7. And this is for strategic actions at cell surfaces that are important in innate immunity against bacteria, and also in advancing tumor cell invasion and aggressiveness. And that we've been able to use PREs to detect the transient proximities to spin-labeled partners, such as bicelles and glycosaminoglycan chains. And that these, in fact, bind the same surface on the back of the catalytic domain. And we found that, that is, NMR found that 30% of the top in silico hits are binding at a serendipitous site. And we could rank these ligands and fit their affinities efficiently, actually without needing any peak assignments or anything. And we found that 10% of the binders inhibit, and that actually the majority of them turn out to be allosteric activators. And I'd like to thank the very productive and creative postdocs who did this work, Steve Pryor and Jan Fulcher. She's come back, and she and Rama have worked on the drug discovery, and Denzel Bernard did the virtual screening in San Francisco at Atomwise. And these are Bob Linhart and Fumin Zhang, who are instrumental in the GAG part of the story. And I'd like to thank our sponsors and you for your attention.
Thank you, Steve. You too, bang on, right on time. Very good job.
I have a question, Walter.
Please.
So that was really nice, Steve. So you did really beautiful studies showing both GAG binding and membrane binding. And one of the things that membrane people often forget is that, you know, the outer surface of the plasma membrane has a complicated extracellular matrix attached there.
Indeed.
The GAG is a part of.
Yes.
Have you done any modeling just to see if you have the protein both docked to the membrane surface, if it then makes sense where the GAG sites are with respect to that?
Well, as I mentioned, when it comes to anionic membranes, which could include lipid rafts, then there's going to be a steric clash that both the GAG, at least part of the GAG binding, and the principal membrane binding site coincide. So they can't both bind there, but the GAGs could bind elsewhere on flanking sides of the catalytic domain. I think on the cell surface, we'll mainly see the catalytic domain, you know, the active form without the prodomain. I think that if we had the zymogen on the cell surface, which may be just a transient phenomenon, then yes, they can both coincide, or they can both be concurrent, at least transiently, And I suspect, Chuck, that the glycosaminoglycans really draw the soluble enzyme in toward the cell surface and promote its activation, and then it may settle down in a more non-specific fashion elsewhere on the bilayers. When we did some live cell imaging, interestingly to me, we saw a punctate pattern of fluorescent staining to the cell surfaces in the case of this enzyme. And so it has definitely some tightly preferred locations of binding. And I have to wonder if those are some sort of heparin sulfate proteoglycan containing assemblies that they're localizing at.
Yeah, thank you. That's great.
There's a question in the chat box that I'll read.
Thank you.
Gajendra Singh. One molecule unexpectedly bound at a different region of the protein. Why is this happening? Are there multiple binding sites?
I think there's only one binding site, and it's not the one that we targeted. The pocket that we targeted is very shallow. Let's see. Okay, the pocket we targeted is right here. Can you see that where the orange cursor is in the lower? This is a rather shallow pocket, and the GAG chain fits over it. This, where we're getting the biggest of the CSPs for all 22 of these compounds is at least 15 angstroms distant, and it's a bigger pocket actually. And actually in a protein science graduate course that we were teaching, one of my co-instructors was teaching that in these Tim barrel proteins, this certain motif, which involves a beta strand and a turn and a helix, is always an active site in the Tim barrel proteins. And that corresponds to this site in this enzyme, interestingly to me. So in Tim barrel proteins, this is an active site. Not that they're related to MMPs, but yeah, I don't think it actually binds over here. I mean, we do have a very few CSPs in this loop above that site, and if there is binding, it's even worse than the binding over here.
So Steve, if I may have a follow-up quick on this, what made you think or target the first binding site, and would there be any contradiction with the now newly found binding site?
Well, I actually got this idea from Bill Parks, a famous pathologist who's at Cedars-Sinai in UCLA. I got this idea about 12 years ago. He thought, well, we could have a potentially good therapeutic if we could prevent the binding and activation of this enzyme. And so since then, I've been wondering if we could prevent the binding of the GAGs. And so I just wanted to outcompete that GAG by doing the red X's that are shown in this slide. And so that was the strategy, and actually independent of my desire to outcompete it, this site over here is the site that Denzel Bernard chose as suitable for that purpose. And so he thought it was a reasonable pocket, but you know, it's a little bit like trying to drug a protein-protein interface, which is so flat. And those are sort of infamously challenging to target. And so I think it's a similar situation here. It's just kind of too flat and shallow. And so it was maybe less likely to succeed.
But it was kind of hypothetical from the beginning, not based on other data.
Yeah, but you know, I was an optimist, and I guess they were too. So
thank you.
Do the two sites have similar characteristics, electrostatics, or?
They both are, they both have a lot of positive charge around them, Walter. These two are both basic residues.
Yeah.
Actually, we're looking end-on the binding pose of the 8-mer heparin chain that's bridging from the pro-domain in the background in red up into the catalytic domain. And what's likely happening with actual heparinoid chains of physiological length, sort of the size of low molecular weight heparin on up, we're talking like 16 mers on upward from that. It's likely that they are, in my opinion, I would propose or hypothesize that they actually wrap around this enzyme. Like 50% to 70% of the way around this enzyme, and that they actually, this is right on the trajectory to go from this binding mode to the binding mode that's not pictured here, but which is shown over here. So if you just draw a line between these, this chain is more or less going over the site where these compounds are binding. Now, how tightly it adheres, I don't know, and it's interesting to me that most of the compounds actually promote activation, but two of them, I guess, present enough bulk in the right place that it somehow interferes in the GAG interactions and the promotion of this activation, which is something that occurs by proximity. The GAGs bring these enzymes into a clump that can activate one another in this clump, in this aggregate.
Okay.
So the GAGs may be breaking that up.
We have time for one very quick, simple question.
Yeah.
You said that you were using heparin-8. How do you, was that synthesized, or how do you separate it from a mixture of other heparins?
Well, we don't. Bob Linhart and his group are very good at that. They use HPLC, and so they simply fractionate heparin that they buy, and then they can manipulate it.
Yeah. Okay.
Thank you very much.
We're right on time again. And so now I'd like to introduce our fourth speaker, who is Muriel Maurer. She's from the University of Louisville, and her lineage traces to Harold Chiraga, another one of the giants, maybe not so familiar to all of you, but someone who studied a great deal NMR in combination with computational analysis, and is stayed active well into his 80s. Muriel.
All right, are you able to see the slides?
All good, see and hear.
Okay. All right. Well, I'd like to thank the organizers for the opportunity to make this presentation that's associated with the thrombin exocytes and their maturation. There are perhaps few people who know that actually Harold Chiraga, very early in his career, actually did do work that was thrombin and fibrinogen related. Thrombin is part of the blood coagulation cascade. It's a cascade that has a variety of zymogen proteins which need to be activated, and each case it might be like a factor 11 gets activated to become a factor 11A. You have two different pathways that you can do in order to get your way across. Eventually you branch out and you get to a common point here where you have activated factor 10A. And then that is responsible as part of the prothrombinase complex to convert a protein which is known as prothrombin over to an active serine protease, which is known as thrombin. And then thrombin is then involved in allowing you to have conversion of fibrinogen to fibrin. You have the formation of a fibrin clot, which then is covalently cross-linked with factor 13. And in addition, there are going to be red blood cells and platelets that are present in that particular group. It turns out that thrombin does not just have these particular functions, but also has a variety of other functions. It activates a number of the coagulation factors that are present. It involves in procoagulation activity, conversion of fibrinogen to fibrin. It helps to activate protease activated receptors that are involved in platelet activation and eventually into inflammation. And it binds GP1B alpha, an enzyme that actually helps to promote the protease activated receptor activation. It also has some anticoagulant functions that are present, in which it can activate protein C in the presence of the cofactor thrombomodulin. So it has all of these different activities, and it needs to be highly regulated in how those processes and activities are going to take place. So what this serine protease thrombin has is that in addition to its catalytic site and a series of surface loops, it also has what are known as anion binding exocytes that are present. They have a number of basic residues that are associated with them, along with some hydrophobic aromatic character that are present. And thrombin utilizes these exocytes to help to tune some of its actions. A lot of times it helps you to orient a substrate properly so it'll work well within the catalytic site. Other times it might be involved in cofactor recruitment, being assured that, say, something binds with an anion-binding exosites 2, being assured that the thrombin can be well poised to do its next particular functions. If we look in terms of the anion-binding exosites that might be present, the ones that I'll be talking about are the ones associated both with the PAR1 and also with the PAR3 groups that are over here. And then in terms of examples of ligands that are AB2 related, heparin is one that commonly goes to this site, and I'll be looking at one that has to do with the GP1B alpha group that is here. So if we want to look at thrombin, we want to think that it actually appears originally as a prothrombin group, here as a zymogen prothrombin, and it has immature exosites that are present. For the AB2 site, it has kringles and actually a glaudomain, which is not visible by X-ray crystallography, that blocks this site, and they're both immature. You can convert prothrombin over into prethrombin 2. The glaudomains and kringles are gone, you're still not fully mature, and then eventually when you get to the thrombin, your active site area is exposed, and your 2 exosites are now available for their particular types of functions. There had been interest in trying to figure out, is there any possibility that you could get any ligands to bind within these exosite regions ahead of time before you're fully mature? And also kind of a better understanding of what really happens when you try to do some exosite maturation. So the studies that I'll be talking about are ones in which we kind of consider this conversion. Is there a way that we can monitor how you can go from a immature proexocyte to a mature one? And can you look at particular ligands that might be able to bind to those 2 particular types of regions? And a region of concern is the fact that according to X-ray crystallography, there really isn't a lot of difference along the areas of some of the anion-binding exosite regions. This is in terms of the 70S loop, which would suggest from X-ray crystallography that there are not really big differences across these units. The other thing that we want to look at is then to think about if we place something at anion-binding exosite 2, will we be able to see some inter-exosite communication over to the anion-binding exosite 1 area. So these studies are going to be done by NMR approaches, allows us to look at binding affinities, allows us to look at changes in chemical environment, and allows us to look at this from a single amino acid perspective. Initially, you would probably think if we're going to look at prothrombin and thrombin, that's probably the enzymes that would be isotopically labeled, but there's kind of an issue to kind of think about for the thrombin molecule. This is a spectra taken from Betsy Combovez's group at UC San Diego, and thrombin is a highly dynamic molecule, and it turns out that when it doesn't have any ligands bound at all, there are a number of areas for which chemical shift assignments are difficult to make. In this particular case, it is, has a PPAC, a small molecule at the inhibiting the active site region, but there are still stretches for which you cannot make chemical shift assignments. And for this particular project, the area within the 70S loop and the 30S loop are still sections for which there is difficulty making the chemical shift assignments. And we wanted to be able to probe specifically within these regions. So what we decided instead was to sort of flip the project and look at it from a little different perspective. The enzymes are going to be unlabeled, and it's going to be our peptide ligands, which are going to be N15 labeled. And so we're going to be doing HSQC NMR titration studies that I'll talk about, and all of the work was done by graduate student Ramya Biar when she was in my group. And the studies are then done with unlabeled prothrombin, unlabeled thrombin. They're done in excess, and the peptide is then kept at a constant concentration. And then there are serial dilutions that are then done to reduce the thrombin or prothrombin down while maintaining a common peptide concentration. And then you can look at the binding of the protein to the peptide. And then it will be screened from both a 1D and a 2D perspective. We'll look to see, do we have fast exchange that takes place across where we have a free state, a bound state, and weighted contributions that come across? Are we in slow exchange, or are we in an intermediate exchange where we'll have difficulty in terms of assessing what those KD values are going to be? Okay. So in order to first start out with this project, we wanted to first do some one-dimensional protein line broadening just to decide, are we at least going to be able to work with these peptides? These are done the more traditional way where you have an excess of peptide and a smaller amount of the enzyme. And we had an initial sequence that we were looking at for the PAR3. original for the PAR3 group. And in that particular case, we were first looking at the original sequence that was then present. And there we can see what the free peptide looks like. We can see that there already is line broadening that takes place in the presence of the prothrombin, which suggests to us that a PAR3 peptide already has the capability of doing the binding. And then when you do it in the presence of the thrombin, there's even greater bounding that takes place. The studies were first done with just the proline being present here, but it seemed like it was going to be too tight of a binding. So all the studies that I'll show you will have actually a glycine that is present, which makes it a little bit weaker, but still allows us to do all of the screening studies. Okay. For these particular work, Ramya would have N15 labeled, we'd have N15 labeled peptides prepared, and we'll look at them at specific regions that are going to be N15 labeled. And we'll start out with an aspartic acid D54, which makes a salt bridge over here to the thrombin R77, and then also a phenylalanine 47, which makes pi stacking interactions over to the F34. And then we'll look to see what happens in the presence of the prothrombin with the immature exocytes versus the thrombin with the mature exocytes. Okay. And so in each case, the way Ramya would do this is she would start out with her complex, she would do a 1D spectrum and then a 2D spectra, then do a serial dilution and then continue her way through the project. So you can see, so she started out here, she's got a 2-to-1 ratio of prothrombin to thrombin. She barely sees any kind of peak at all, definitely suggesting to her that this PAR3 peptide already has ability to interact with the immature proexocyte. And then as she dilutes down the enzyme, then she's going to see eventually the free peptide. If she looks at the thrombin case, then she sees even more extensive broadening. It's probably now in a more mature state, and she indeed is going to look at that. We can look at it from a two-dimensional panel perspective, where she can look at, let's say, the phenylalanine 47, where she can then do see small changes in chemical shift with the immature prothrombin. When she switches it over to the PPAC-inhibited, active site-inhibited thrombin, she'll see a bigger change in chemical shift. The F47 must see a new chemical environment in the presence of the thrombin with the mature exocyte. If she looks at the D54 group that's here, she can start at 2-to-1 and go across. But in the mature thrombin case, she can only see something at a ratio of thrombin to a peptide of 0.16 to 1. She's definitely probably trapped in some sort of intermediate exchange area where she cannot see the peaks very well. But indeed, it's going to be probably a tighter interaction. Okay. And then, so she is probably, in the case of the D54, probably caught more intermediate exchange. In the case of the F47, maybe more likely to be in the fast exchange condition. She can do the individual KD calculations for that and then show that the F47 in the prothrombin state has a KD of around 65 micromolar. If she then converts it over to the thrombin case, then she has more of KD, more about 40 micromolar, but she sees a bigger change in the chemical environment that she's looking at. If you look at the D54 case, there it is a situation in which she has got the same, relatively the same KD in the prothrombin case, but upon activation to the thrombin, she ends up with something which is much too tight for her to be able to figure out a KD value. Okay. So the next strategy then to look at was then to see whether or not she could look a little bit further at these types of interactions, and particularly what would happen if she were to disrupt this salt bridge that takes place between the D54 and the arginine 77. Would she then have the capability, is it just going to affect this region, or is it going to affect longer also over into this particular region over here? And so far we already know that there has been some capability to interact with the prothrombin, the immature one, and with the mature thrombin. So in this particular case, we'll just be looking at the thrombin case that is then here. And she is then able to see that she definitely can start to see these peaks earlier in her titration already across. And then she can very nicely see that she has nice changes in chemical shift that takes place as it encounters that new environment in the mature thrombin. And she has then KD values, which are then significantly weaker than what she had before. So both amino acids are encountering a new environment. That salt bridge was probably important for the D54, but it also is influencing individually what's happening at the phenylalanine position also. Since that strategy seemed to work well, we then thought, okay, let's have her try some other ideas. Again, we'll look at something that has electrostatic component capability that's over here, with a glutamic acid 48, and then also a leucine leucine 52 residue, which also has capabilities of looking at some hydrophobic residues that are present in the thrombin molecule. Again, we'll compare the prothrombin to what happens in the case of the thrombin. Okay. In this particular case, if she, as Ramya was looking along, in this case, she can see at, at ratios of 4:1 of prothrombin to peptide, she's already can see the peaks. That already gives us a sense that she probably has a weaker interaction that's taking place. And then in the PPAC thrombin case, they then become much more broader already at the start of the spectrum, indicating that she has something that's tighter. In these particular cases, what she then has is that she can look in the patterns that are over here for the prothrombin, and she doesn't see as bigger changes in the chemical shift that takes place. If she goes over to the PPAC thrombin, she does have cases where she's seeing greater changes in chemical shift upon the capability of going to the active thrombin molecule, but she's also got these cases where she is definitely tighter. It's going to be a little bit harder for her to figure out what the KDs are going to be. And if she does look at those, the E48 is going to be a little bit tighter in its binding than the L52. So again, a sense that those electrostatic interactions are binding a little bit tighter than what she's seeing from the hydrophobic interactions. If we look at this overall in terms of what she's been able to look at and the different residues that she's been able to see, she's been able to look at a phenylalanine 47 and a aspartate D54 in the presence of prothrombin. Both cases, they had a KDs kind of around the 65 or so micromolar level. Upon the conversion to thrombin, the F47 is a little bit tighter, but what it really has is a much greater change in chemical shift that it notices a new chemical environment. The D54 doesn't notice that as much, but it has a much tighter affinity and much tighter binding that takes place. If we were to remove the salt bridge contacts that are present here, then both individual residues are going to be impacted by that particular change. They both become weaker that is present. If she then picks another two ones that are associated with the leucine 52 and the glutamic acid 48, both cases, those were weaker that took place and in the presence for the prothrombin, but when she gets over to the thrombin, the effects are sort of similar. You're both at sort of around 40 micromolar or so. And again, something which is acidic leads to something which has a much tighter interaction. So overall, what we can say from these particular groups is she's looking at these PAR3-based peptides. They already have the capability of binding to prothrombin, something that has an immature exosite that is present. There's probably some sort of binding competent population that is present already within the prothrombin case. Once she gets over to the thrombin case, there's a full formation of the mature exosite. You must more dominate that nice binding conformation, and she's able to see more of them binding. She's able to see actually tighter binding. And then what becomes kind of nice with this particular project is that she's really looking at this at the individual amino acid level, watching how individual amino acids are responding to whether you see an immature exosite or a mature exosite. And these types of changes were not visible by just comparing these types of surface loop areas through X-ray crystallography. Based upon the successes with the PAR3 peptide, we then said, oh, let's also try and see what is going to happen in the PAR1 case. The PAR1 protease activated receptor is actually an example of one which is more commonly viewed in terms of this particular family. Again, there's an X-ray crystal structure which is associated with that. And in this PAR1 case, from X-ray crystallography, there's a segment that mutagenesis studies had suggested would be key for a binding contact area, but in X-ray crystallography, that area is somewhat disordered. And the strategy was then going to be the same. If you take a PAR1 peptide and you look at individual types of contacts, what will you be able to see in terms of the environments? In this particular case, we first had done some experiments with it with the proline that was present. We knew that we could get the PAR1 with the proline to bind to the immature prothrombin, but we wanted to introduce a glycine similar to what had been done with the PAR3. And in that particular case, we ended up with a peptide that really doesn't have much interactions with the immature prothrombin that is present. So it must not be that all peptide sequences have the capability of nicely interacting with the prothrombin. But we still wanted to see what could we see from the thrombin perspective and what could we sort of map out in terms of sites of contact. The aspartic acid here at D50 had a very solvent exposed amide proton, which made it hard for her to look at this one, but she was then able to look at this F55 and see a nice changes in chemical shift, new environments that phenylalanine 55 saw when it was in contact with the mature anion binding exosite 1. and it gives kind of a, a weaker kind of KD that's associated with that. Okay. She can then look at other ones. She can look at a glutamic acid that is present here at 53, and then in a aspartate at 58, which would allow her to look at a region that had been difficult to see by X-ray crystallography. The E53 does not have much contact here with a prothrombin, and in the case of the PPAC thrombin, it also does not have relatively weak interactions that take place. By contrast, the D58 does see nice changes in chemical shift. It does then able to experience a new environment, and she can get a KD values that are present, something again, the idea that we see something sort of in the 40 micromolar type of range. She did other ones. She did a lysine, and she also did a glutamic acid that was present, and they were both examples of ones that were a little bit weaker in terms of what their KD values were. But instead, she was still able to monitor across sort of regions that might be important, and that this D58 was indeed probably a key anchoring site to take place. Okay. And if she looked at these kind of across, and she did a number of them there, she could both look at electrostatic interactions, also look at hydrophobic interactions. The interactions of the sequence that we chose did not have as extensive interactions with the immature prothrombin. Her best context to look at D58, which probably is involved in some sort of electrostatic interactions with something on the thrombin surface, and also the phenylalanine 55, which similar to the PAR3 also had similar context within this particular type of a region that was over here. So the next thing that we wanted to do was we've been exploring these interactions that take place, whether we're with an immature exocyte or we're looking at a mature exocyte, but then we wanted to also consider the possibility of inter-exocyte type of interactions. And so in that particular case, thrombin has these two anion-binding exocytes that are present. And in my research group, we had done some extensive studies involving hydrogen deuterium exchange coupled with mass spectrometry, where we placed ligands at AB1 and then looked to see what kind of regions of solvent protection or solvent exposure could take place if you put ligands at AB1. Do you have local? Do you have longer range changes that might take place? The same thing was done at anion-binding exocyte 2 to look for local and long-range effects, and also to place a ligands at AB2 and then see what impact it would have on the binding of ligands at AB1 region that was over here. So we thought in these particular studies, it is examples for which we could, based upon how we did them, we could only really look at stretches. So stretches of AB1 or AB2 or other portions of the enzyme that were affected. And we thought these NMR titrations might allow us to also be able to look at individual amino acids to see how they might respond. If we place something at AB2, how well will it interact with the mature exocyte on AB1? So for a ligand for the AB2 site, we picked GP1B alpha, a peptide sequence that's based upon that. And physiologically, the GP1B alpha will bind here to the AB2 region of thrombin, make it so the thrombin is better able to place PAR1 region on here so it can be activated. The GP1 region that binds to the anion binding exocyte 2 has a number of acidic residues that are present here, and has typically sulfated tyrosines that are present. And in our case, we substituted them with phosphorylated groups. A thrombin is prepared together with the tight binding GP1B alpha peptide, and then experiments are then done with peptides based upon PAR3 and also based upon PAR1. And then we'll look to see, are there any tightening or weakening that might take place to promote inter-exocyte communication. Okay. So we can start out first with the PAR1 case that is present, and each time these are with the thrombin molecule. We can look again. This is one where the E53 group was too weak to get good KD measurements. The D58, on the other hand, was the one that had the nice change in chemical shift and where you had something at around 40 micromolar. If we take the E53 and we do the experiment where Ramya has taken PPAC inhibited thrombin with the GP1B alpha that's present, and she's doing the titration with the PAR1, she sees that when she couldn't really get a KD here, now she can get a KD at around 126 micromolar. In the case of the D58, she had something that was around 40 micromolar, and in the presence of the GP1B alpha, so the GP1B alpha is bound while she's also looking at the PAR1 binding, she doesn't see really that much change in the KD that takes place. So this would sort of seem to suggest that the binding of GP1B alpha has some effect on the binding of the E53. It seems to have less effect on the D58. Maybe the D58 is already relatively well oriented and in terms of its interactions. We then wanted to see what happens if we do the same thing with the PAR3. The PAR3 was the one that had worked quite well for us to do these particular types of studies. And so again, she has the active site inhibited thrombin together with the GP1B alpha, and she's trying to do the titrations with the PAR3 ligands. She sees really extensive line broadening that takes place, giving us a sense, well, I'm not sure we're really going to be able to figure out any KDs that are here, but we can indeed say that they're probably strong. And in the case we picked here, the leucine 52, that had been originally something around 50 micromolar in the presence of GP1B alpha. She's now too high affinity to calculate. In the case of the E48 group, she at least has a little bit more that she could be working with, but she still doesn't have enough to comfortably figure out a KD, but is in probably even tighter than what she had seen before. So in this particular case, she's able to see that in the presence of the GP1B alpha, the PAR3 related peptides, 2 of them are now benefiting by having the presence of the GP1B alpha.
2 minutes.
Okay. All right, now we're here at the kind of conclusion slide then to look at. And that is then to say that this has been a project to look at exocyte development and inter-exocyte interactions. We focused on peptides based upon PAR1 and also peptides based upon PAR3 that is present, and have looked initially to see whether or not these peptides have the capability to already interact with prothrombin with its immature exocyte versus the thrombin molecule with its mature exocyte, and these peptides already have the capability of doing it. There must be populations already within prothrombin that can accommodate these peptides. Upon activation to the fully activated forms of the enzymes, there are often cases where there is tighter binding, and there are environments where there's greater context, new environments that then take place. And one can also study inter-exocyte communication by placing ligands at ABI ion binding exocyte 2, and they will then inhibit to different extents unique amino acid positions at AB1. So probably a combination of exocyte maturation and sort of long-range allostery is what allows the AB1 conformation to effectively bind to the PAR ligands. All right, and all of this work was then done by Ramya Biyar, a graduate student in my laboratory who spent many a long time doing these NMR experiments. This is from a time when we could still stand together, yes, and like to thank people who've helped us with NMR projects in Louisville, Enrico Teixeira for thrombin mutants, and ongoing thrombin NMR discussions with Betsy Kamabes and funding from the NIH. And,
Thank you. Thank you, Muriel. We already have a question queued up from Angela Dronenborn. And she's just going to ask it directly since she's here on the panel.
For your first study where you looked at PAR3 and you substituted the proline with a glycine, now I noticed that the amino acids used for monitoring are on either side of that glycine or proline. So in the glycine, you have a much larger conformational space that can be sampled by that peptide than if you would have a proline there. You mentioned that the proline binds tighter, but it wasn't clear to me where that information came from.
Oh, that was in terms of the initial experiments that Ramya had tried to do with the proline. She realized that she really couldn't get enough ability to look at the cross peaks. She couldn't see enough of them. She was too much in the more intermediate exchange type of regime. And in order for her to be able to have more points that she could look at, we then opted to make it weaker with the glycine. But looking at carbon-13 based HSQC spectra, we didn't think that we had made any big changes. And in terms of looking at changes, the trends for the changes in chemical shift that she saw were still the same directions, whether there was a glycine there or a proline.
Thank you.
Okay, we have a question in the chat. This is from Gajendra Singh. Nice talk. Do you think that dynamics of the audiolysis loop regulate ABE1 and ABE2 binding, specifically ABE1?
whether autolysis has of the loop has an effect.
Yeah.
I think that there are examples where people know that there is some communication from the antibinding exocytes over towards the region near where those autolysis loops are. So I think that those are important regions to also consider those interactions between those areas.
I actually had a mild technical question. So you report these different KDs that you measure by residue type or for very specific residues. But how do these relate to the actual binding affinities that you would measure, let's say, quote unquote, macroscopically, or for the peptide as a whole with, say, ITC or something like that?
In that particular case, they were sort of in the sort of 5 to 10 micromolar type of range. So I think our average tightest ones were kind of around 30 to 40. But in some of those, we're at least sort of kind of in the same range, but we are not exactly the same.
Yeah, so, but it wouldn't achieve then the, you know, if you used multivalency, you would have very high affinity if you use that simple theory, but it's obviously not like that in this case. Steve, why don't you just ask your question?
Yeah, Muriel, that's very interesting. I'm curious if you see any analogy in modes of heparin binding to the modes of PAR1 and PAR3 binding.
Oh,
to thrombin or pre-thrombin, prothrombin?
We have not done anything with the heparin. We've looked at, and we have not yet studied extensively. I think the most at the GP1B alpha, we have only done some preliminary work. The project that's been done so far makes it exciting to want to try and look at ligands that are along the AB2 region.
New gateway collaboration.
Okay. Yeah, we don't have good sources of a small molecule that's heparin related, so that would be quite attractive.
Well, I'm sure that Bob Linhart and Fuming Zhang would be more than happy to work with you. They seem to relish structural work and NMR work.
Thank you.
Excellent. So I'd like to ask everyone in joining me in thanking our speakers, and we'll close this session. We have a short break, and I don't know, unless Marcus has something to say, we will meet in 15 minutes.